Hello, my name is Henry Hardart, and my presentation will cover intersectional labeling using mouse genetics and how this versatile technique was used to characterize the developmental origins of the various serotonergic nuclei of the neuromodulatory system. Because of how different the organism can appear at different developmental time points, a very logical question to ask in developmental biology is to take a given cell population at a young developmental age and ask what those developmental cells become as the organism becomes an adult. Or, conversely, you could take a population of adult cells and ask which developmental cells those came from. In order to ask and answer these questions, it is important to be able to label certain cell populations based on their developmental origins or their adult fate. There are many ways to label cells, including dye injections, in utero electroporation, retroviral labeling, tritiated thymidine treatment, and other labeling strategies. But these labeling strategies are far from perfect. Some of these labels become weaker as the cell divide, and others lack the precision required to exclusively label certain cell populations. One solution to these problems is to use transgenic mouse lines, or genetically modified mice that are able to selectively express labels in desired cell populations. Transgenic lines are produced by crossing a reporter mouse with a driver mouse. The reporter mouse has a promoter that is always turned on, followed by a lock-stop-lock sequence, and then some sort of label, such as GFP. The driver line has a cell-type-specific promoter that controls the expression of a recombinase, such as Cre recombinase, which is able to cut out anything between two target sites, or lock sites in the case of Cre. The result of this cross is a mouse that has both the reporter and the driver transgene. This means that while most cells will remain unlabeled due to the stop sequence before GFP, cells that express the cell-type-specific promoter will express the Cre recombinase, which will remove the stop sequence, allowing the label to be expressed and labeling the cell. This basic concept can be further elaborated to achieve more specific and thorough labeling of cells, as exemplified by the intersectional labeling of Dimecki et al., in which the investigators separately labeled adult serotonergic neurons based on which rhombomer they came from during development. The serotonergic nuclei of the brainstem are important components of the neuromodulatory system, which has widespread effects on wakefulness, awareness, and autonomic function. In order to label these nuclei by developmental origin, the investigators used one reporter line and two different driver lines. The reporter line has a stop sequence surrounded by FRT sites, followed by a fluorophore flanked by lock sites, and then finally a second fluorophore after the first. Then, the two drivers' lines contain the flip recombinase, which removes items surrounded by FRT sites, and the Cre recombinase, which removes lock sites. This allows the separate labeling of cells based on whether they express only one or both recombinases. In this specific experimental setup, the researchers inserted the flip recombinase under the control of EPET1, which is a promoter which is only active in serotonergic neurons, and placed the Cre recombinase under the control of a promoter which is only active in one of the rhombomeres, for example, EN1 for rhombomer 1. This means that serotonergic neurons from the rhombomer of interest will express both flip and Cre and fluoresce red, while the serotonergic neurons from other origins will only express flip and fluoresce green. This image further illustrates how the intersectional labeling scheme works. You can see that you only get red fluorescence at the intersection of Cre and flip expression. The figure at the top left shows a population of serotonergic neurons that are subdivided into the red serotonergic neurons that came from R1 and the green serotonergic neurons that came from other rhombomeres. The figure in the bottom left shows the primary result of Dimecki's paper, which is the comprehensive mapping of the serotonergic nuclei based on which rhombomer they came from. Based on these results, the authors were able to determine which nuclei are developmentally related, which may lend some insight into the function and relations between these neuromodulatory centers. Thank you very much for your time, and I hope you learned something about lineage tracing and intersectional labeling.